Thank you everyone for joining us today for today's SeqSpace webinar. Today's webinar is from Dr. Christina Curtis and is about next generation sequencing and tumor initiation and progression. Before I pass the microphone over to Tabitha Harrison, who will introduce our speaker, I will quickly review some webinar logistics. All lines during this webinar will be in listen-only mode. You may submit your questions and comments at any time using the chat panel and selecting all panelists. And Tabitha Harrison will ask your question on your behalf during the Q&A session. You may need to activate the appropriate panel using the menu option found at the bottom of your screen, as shown in this image. Closed captioning is available as a link in the chat box, and note that this webinar is being recorded. And with that, I'm going to pass the microphone over to Tabitha Harrison, who will introduce our speaker.
Thank you, Leah. We're very pleased to have Dr. Christina Curtis with us today. She's an associate professor in the Department of Genetics and the Department of Medicine and Oncology within the Stanford School of Medicine. Here, she leads the Cancer Computational and Systems Biology group. Additionally, Dr. Curtis is a co-director of the Molecular Tumor Board at the Stanford Cancer Institute. Her laboratory is focusing on the development and application of innovative experimental, computational, and analytical approaches to improve diagnosis, treatment, and early detection of cancer. Today, Dr. Curtis will present on next-generation sequencing and on tumor initiation and progression. So whenever you're ready to begin, Christina, go ahead and start.
Christina, can you hear us?
I'm not seeing the slides, and I'm not able to hear Christina. Christina, are you there?
Perfect. Hopefully folks can see my slides.
Yes, they look good.
Okay, great. Thanks. Sorry. Great. So it's a pleasure to be here and share some of our recent work on modeling tumor evolution through therapy and metastasis. And really, I'll walk through sort of two different vignettes. The first really takes an evolutionary and population genetics approach to quantify tumor evolution. And I'll then go on and switch gears and talk about a very different strategy using a much larger cohort and really population level data to quantify the dynamics of breast cancer relapse and to elucidate its drivers. So my lab has had a long-standing interest in understanding how tumors evolve. And over the years, we've approached this through a variety of techniques, but really fundamental to the approach that we've used, we have tried to model the process starting from a normal cell, moving on to, in some cases, precancerous lesions, and that's really ongoing work that's taking place now, and thinking about the evolutionary trajectory of a tumor by looking backwards in time at its past proliferative history. And to do this, we've really tried to simulate how a tumor grows in 3D. And so we've built spatially resolved models in silico. These are agent-based models of how a tumor grows, and we can keep track of the phylogenetic relationship between individual cells in this population over time. And then really crucial to this approach is that we've coupled it with techniques from population genetics that allow us to compare the evolution of this tumor and to simulate under these scenarios where we then use robust statistical inference techniques such as approximate Bayesian computing to compare the observed parameters to those in our simulation models. And so I'm not going to spend a great deal of time on the specifics of these models. We've published on this in numerous settings, but just to say that we think that this data-driven approach is really powerful, particularly given that we have no ground truth data really on how tumors evolve in the human system. So today I'll focus heavily on trying to understand the timing of metastatic progression. And we believe that this is an important topic. It's certainly had renewed interest in recent years. This idea of understanding both the drivers and the dynamics of the process has really been revisited with the wealth of genomic sequence data. And I just want to point out that, of course, for many years, many decades, in fact, it's been assumed that metastasis is a relatively late stage event that may take place perhaps after initial phase of treatment, where this genetically advanced subclone then acquires the sort of requisite changes that allow it to colonize a distant site. And really, it's been through next generation sequencing that these kinds of questions have gained renewed interest. So this is one of many recent review articles that have been published that have started to really come back and ask the question as to how this process takes place. So one simple model is the linear evolution model, where mutations are accrued and this genetically advanced cell then goes on and metastasizes. Certainly, there have been other models, such as the notion of early dissemination with parallel evolution at different metastatic sites, i.e. different mutations being accrued along different lineages. There's been models proposed where we have dissemination arising from a single subclone or perhaps from multiple subclones. And so this is just one such example. I'll show another just to maybe spark your interest in this and say that it's not just about the timing, but it's also about the mode of evolution and whether these cells are arising from a monoclonal origin or perhaps a polyclonal origin. And so in this scenario of monoclonal dissemination, we can imagine that it is one effective branch on this phylogenetic tree that gives rise to the metastatic clone. In an alternate model, we could imagine a polyclonal tumor where in fact the tumor, the metastatic clones arise from, again, different branches, but at the same sort of a monophyletic mode from the primary tumor tree. Or we could have the scenario where, again, polyclonal dissemination, but arising from a polyphyletic mode. And so this is complicated, and there's a lot of questions about whether we actually have the data we need to resolve this. My group's been interested in this question for some time, and to really address this question, we compiled a cohort that consists of a number of patients that had primary colorectal cancer with distant metastasis to either the liver or the brain. And for each patient, we were very careful to obtain multiple regions from each sector of the tumor, and that really was based on our prior work looking at heterogeneity in primary colorectal tumors, where we found that indeed just spatially sampling from small regions apart of the same tumor gave rise to patterns of dramatic diversity. So we knew this was essential. And in this cohort, I want to point out that we also were very fortunate to collect samples that really span the continuum of disease. So liver exclusive metastasis occurs in about a third of metastatic colorectal cancer patients. Oftentimes, it is treated with curative intent by surgical resection of the liver metastasis. In contrast, brain metastases account for a mere 4% of metastatic colorectal cancers, and really brain exclusive metastases are even far more rare. And so we reasoned that perhaps these different patterns of distant metastasis may be associated with different evolutionary trajectories that reflect their aggressiveness. So one of the first analyses that we did with this cohort was to go back and ask the question, what kind of alterations do we observe? Just taking the list of canonical alterations defined by the Cancer Genome Atlas. And so I'll point out that for each patient shown here, separated along the vertical, we have multiple regions per primary tumor denoted by a P, or lymph node metastasis, or brain metastasis. And so we've amalgamated all of the different sequencing regions for this summary plot here. And what I hope you'll appreciate if you look across is that, of course, canonical alterations such as APC, TP53, KRAS, or SINE1 are found in almost all regions of a tumor from a given patient. They tend to be quite prevalent. And as we sort of go down the list, we can look at more rare events. This is true, by the way, for both the brain metastasis cohort and the liver metastasis cohort. It is also true if we look at the copy number level, although not shown here. Now, another way to summarize these alterations is to ask the question, where are the vast majority of mutations occurring? So in this simple representation of a phylogenetic tree, what we're showing is the alterations that occur on the trunk versus those that are unique to the primary lineage or unique to the metastatic lineage. And what you can see quite dramatically here, where we've just counted these events for each patient in the brain met cohort or the liver met cohort, is that the vast majority of alterations take place on the trunk of the phylogenetic tree. And this is in fact consistent with earlier results from our own group looking at the prevalence of somatic mutations in colorectal cancer. This is in sporadic colorectal cancer, but it's also consistent with earlier data using microsatellite-based markers to read out mutations present in the colon. And this really suggests that the vast majority of these events are taking place prior to transformation. And to look at this yet one more way, we can now take the compendium of colorectal cancer specific drivers and ask where do these drivers occur? And again, what you can see here is that they're dramatically enriched on the trunk of the phylogenetic tree. And in fact, we see very little evidence for enrichment in the primary or the metastatic lineage. And this is in fact true if we look at all drivers from other epithelial tumors or looking pan-cancer. So most of the alterations that are really critical and that we think of in this context are occurring very early. Now, with this relatively unique cohort in hand, where we've sampled multiple regions of the primary and distant metastases, we were also very careful to obtain really comprehensive treatment information. And this is, of course, essential because we know that therapy can dramatically alter the evolutionary landscape. So I'm showing you one example for patient V930. You can track this patient's disease course. They actually had a very long disease course, surviving over 10 years with their disease, following the surgical resection of the liver metastasis. They received chemotherapy, 5-FU. They did have a lung metastasis present at that time that we unfortunately were unable to sample. And then they went on to develop a brain metastasis. And so we have this information for all of the patients in our cohort. What I want to point out is that agnostic to this sort of clinical information that we've collected, we can use the genomic data to reconstruct a phylogenetic tree. And so what I'm showing you here, we can again see on the trunk of the tree that there's a number of key canonical drivers such as APC, KRAS, SINE1, and TCF7L2. The primary lineage shown up here in green forms really a monophyletic clade, and we can in fact see just from the sequence data, looking at these patterns of divergence, that the brain metastasis appears to have descended or arisen from the lung metastasis. And so looking at these trees gives us some clue about the relative divergence times based on molecular data. And we can, in fact, use this information to infer the likely course of disease spread. And we do this many times over. And what you'll see here is that our inferred patterns of dissemination match very well with what we know clinically about this patient's disease course. So this is all very nice, and in some ways, it's quite exciting that we can leverage these patterns of diversity and sort of evolutionary principles to start to uncover the routes of systemic spread. On the other hand, I want to point out that there's really fundamental limitations to just using a phylogenetic approach alone. And one of the challenges here is that we're tasked with taking bulk genomic sequencing, in this case it was exome sequencing, to trace back this minor subclone that was the most recent common ancestor of the primary tumor sample. And so this is actually very hard when we're sampling just several bulk regions of the tumor. So even though we've attempted to account for the spatial heterogeneity by performing multi-region sampling, it's absolutely the case that we haven't sampled all cells in the tumor. So this is but one problem that we've tried to address here, by performing multi-region sampling. The other challenges are that there's almost certainly variation in the mutation rate as this tumor evolves. Inherent to the metastatic process is the fact that we have this bottleneck. So it's generally assumed that a small number of cells, perhaps clusters of cells, go on to disseminate, but it's going to be a very strict bottlenecking event that occurs. And so that, of course, is going to skew some of our inferences here. And then finally, and I won't dwell on this too much, but in our past work, we had demonstrated that starting in colorectal cancer, but really with extension to all solid tumors, that there can be at least two dominant modes of tumor evolution. We could have a model that's driven by very stringent selection for specific driver alterations that take over and dominate the population, or we could have a model of effective neutrality. And really, these phylogenetic approaches don't directly account for this. So to overcome these limitations and get back to the question at hand, we developed a computational inference approach that we call SCIMET. The basic idea about this approach is that we are interested in measuring two key parameters from the patient genomic data. We would like to measure the mutation rate, and this is, of course, just at a snapshot in time. We would also like to measure the time of dissemination, and that's relative to the primary tumor size. So to accomplish this task, we really turned back to these spatial agent-based models that we had pioneered in the past. And in this approach, we simulate a tumor comprised of up to billions of cells, and we keep track of their spatial relationship. And the reason for doing so is that we know that space can fundamentally alter clonal evolution and their dynamics. And then we simulate the dissemination process. We can imagine that we have a small number of cells that go on to seed the distant metastasis. And now we have a spatially evolved metastatic lesion, and we can subsample from that. And with this information, I should say within the simulation, we simulate the entire sequencing process and the errors associated with that. We can also simulate differences in tumor purity. So we're really mimicking in this agent-based model the kinds of differences that we see in our experimental approach. So now what we can do is to derive a number of summary statistics from the data. So we measure things like the cancer cell fraction or the variant allele frequency. This is simply a measure of the number of cancer cells that harbor a particular mutation. And we can then compare the simulated data with the observed patient data and compute a number of summaries from this. And so when we apply this approach to this cohort, we can measure these two fundamental parameters. So I'm showing you here the brain metastasis cohort and the liver metastasis cohort. In this heat map, each patient is represented along the horizontal. So we have now the mutation rate, and these values are the posterior probability distributions from this approximate Bayesian computing framework. So what you'll hopefully see here is that from these exonic profiles, we are inferring that there's about an order of magnitude variation in the mutation rate across patients. And in fact, the variation in this rate is pretty comparable between those patients that had a brain metastasis, which we might perceive of as being far more aggressive, and those patients that had a liver metastasis. Now, on the right-hand side, we are instead inferring the ND value, and this is the number of cells that were present in the primary tumor at the time of dissemination. And we really focus here on the size of the tumor because we think that it's more clinically relevant when we consider the imaging modalities. So you can envision that typically for colorectal cancer, we don't start to detect these tumors till they're on the order of about 1 centimeter cube, and that's still relatively early. So what you can start to see here is that for the majority of patients in this cohort, we're finding that the primary tumor was at a size that was as of yet clinically undetectable when those cells disseminated and seeded a distant metastasis. And there's a subset of tumors where we tend to see slightly later dissemination. So these are actually the confidence intervals that you can see here. In fact, we're being very, very conservative with these estimates. So still, these are relatively early. And I think in many respects, this was a surprising finding for us. And I do want to point out, however, that all the patients in this cohort did have a clinically detectable distant metastasis. So we're limited in the sort of denominator. We're looking at all patients that had a clinical metastasis. So now, this is all very nice and suggests that maybe we need to rethink the timing of metastatic dissemination, and that for the very first time, by combining these population genetic approaches with patient genomic data, we've been able to quantitatively estimate this process. However, our sample size was relatively small due to the challenges of obtaining such cohorts. And so what we then did was to ask, well, is there something that we can learn about the fact that many of these mutations are happening back here on the trunk of the phylogenetic tree? And could we take these specific combinations of mutations that seem to be potentially associated with metastatic potential and actually validate that in an independent cohort? So what we did was we turned to targeted sequencing data. This is really based on small panels of known oncogenic drivers, so far more reduced set of genes than from our exome sequencing. And we compared the distribution of these alterations in patients with early stage disease versus those with metastatic. And what we found was fairly striking, namely that if we look at particular combinations, and we call these oncogenic modules of driver events, so in this case, APC in combination with the TP53 and PTPRT mutation, we see that they're almost exclusively found in patients that have metastatic colorectal cancer. The same goes for other combinations such as APC, KRAS, and PTPRT, so you can substitute in KRAS for TP53. But there's also other additional drivers such as AMR1 and TCF7L2. And what these genes and combinations have in common is that they tend to be associated with overcoming niche independence and with Wnt signaling. And so this isn't really necessarily a terrible surprise that if you have the right combination of events, not just any combination of events, that the tumor cell is really poised to disseminate. And one way of thinking about this, obviously there's a number of implications, but really this harkens to the fact that we have this sort of 3 to 4 hit model where by the time you accrue these mutations during pre-malignant evolution, the horse is out of the barn. And this, at that point, the tumor cell may very well have the capacity that it needs to go on and invade and metastasize. So really, these findings harken for the need for earlier detection, for example, using cell-free DNA-based methods. And I would point out that a particular benefit of these alterations that we found is that they tend to occur very early, so they are in fact clonal, making them more readily detectable in the circulating plasma. Now, by the time we typically detect these tumors, once they've already presented and hopefully at an early enough stage, these specific combinations of drivers may serve as biomarkers that in turn inform the approach to adjuvant chemotherapy. And in fact, some of the modules that we defined suggest new therapeutic paradigms. Obviously, another key area of interest is understanding how we can combine these targeted therapies with immunotherapies more rationally. And so we can envision a scenario where, understanding the combinations of events that are present, for example, in a stage 2 patient versus stage 3 may dictate also whether that patient is likely to benefit from adjuvant chemotherapy. And currently that's of great debate in the clinical oncology community. So far I've shown you that at least in colorectal cancer, there is evidence in the human setting for early metastatic dissemination. And this finding really turns some of our assumptions on its head. But I think that there's a number of other challenges that we need to address as a community. And one of those is really expanding the scope of these studies to other tumor types. So there's been great interest in recent years in sequencing metastatic lesions. They're, of course, underrepresented in cohorts such as TCGA. There's been a number of efforts to do so. The latter of these two focused exclusively on sequencing the metastatic site. There was no matched primary. And that's, of course, due to the challenge in obtaining paired samples, sometimes that are obtained decades apart. And there's also been really elegant work that has started to unpack what the impact of therapy is on the cancer genome. And in many instances, this impact is quite profound. And I think given the limitations of sort of treatment annotations, we're actually only at the first steps of beginning to understand how alkylating agents and different types of chemotherapy mutagenize our genomes. So this really led us to expand the scope of this study and to ask, could we compile a cohort of paired primary and metastatic biopsies that would allow us to at least look at the most common epithelial tumors? And so we culled the literature and we really pulled down and analyzed within a uniform bioinformatic framework every paired primary and metastatic sample we could find from colorectal cancer patients, from lung cancer patients, and from breast cancer patients. And what you can see in this Sankey diagram is that These tumors, of course, have different patterns of organotropism, so that's sort of captured here. But we can also see that our cohort was roughly split with respect to untreated versus treated metastases, and that's very important for the analyses that I'll go on to describe. But I do want to say that this actually is quite biased with respect to the tissue of origin. And in fact, some 83% of breast cancer patients in this cohort saw therapy, and that's far greater than the colorectal and lung cancer patients. So one of the first questions we had with this data was, well, what's the prevalence of mutations that are unique to the metastasis? And part of this was motivated by these earlier studies that I showed you, where a number of metastasis-specific drivers were identified that were not found in unmatched primaries from TCGA, for example. And what you can see in this summary plot here, so we're looking at just the mutational landscape for the most common events, and you'll see the frequencies taper off quite dramatically very fast. But we're looking at the prevalence of events that are found that are shared between the primary and metastasis, that are metastasis-private in red, primary-private, and we've further broken this down as to whether they're clonal or subclonal. But what I want to point out here is that actually there's quite a bit of metastasis-specific events that you see on this landscape plot. And in fact, it's far more so than we tend to see in colorectal cancer and in lung cancer. And colorectal cancer cohort includes some of the samples that I described in the previous study. Obviously, we've expanded on that, but again, consistent with what we observed there, in colorectal cancer, there's a ton of sharing of these mutations, and they tend to be truncal, so they're clonal. So why might that be? Well, the reason that we believe that we're seeing more metastasis-private alterations is potentially a function of therapy. And so if we break this down, I'm just showing the proportion of those events. Those that are shared dominate. Those are events that are happening prior to transformation during normal cell division processes, those that are primary specific or those that are metastasis specific. And in fact, if we break this down further, what we can soon see is that this really parses out with the treatment history of the patient. So if we, our numbers are fairly small for the treated cohorts for colorectal and lung, they are statistically significant, but just a caution about the numbers here. In contrast, in breast, we indeed see with far greater sample size that there's an enrichment of metastasis-specific events in patients that received prior therapy. So this is interesting. I think it gives us a cautionary tale about what to infer about new therapeutic targets when we haven't compared with a matched primary. But also, just to really diagram this out in a very simple way, we believe that this also speaks to the nature of how tumors are evolving, and really that we could envision a scenario where we have this major clone present when we are in a scenario where the tumor is treatment naive. This cell goes on to see the metastasis. Most of the drivers are occurring on the trunk of the phylogenetic tree. In contrast, by the time the tumor has seen therapy, we've selected out, in fact, for a potentially resistant clone that is more aggressive and going to fuel seeding of that metastatic lineage. And this indeed will give rise to these patterns that we see where there's mutations on the metastatic lineage. So let's come back to our models of tumor evolution. We talked about this a little bit before. For colorectal cancer, we envision starting with a normal cell. These undergo many decades of precancerous evolution. And I think we're only beginning to appreciate just how very long and latent this can be, and really have a very limited understanding of those clonal dynamics. But clearly, there's absolutely selection and clonal expansion taking place. And then we give rise to this malignant founder cell. And so as we were interested in estimating this timing in colorectal cancer, we similarly sought to measure this TS value. So this is the time of metastatic seeding when the metastasis-founding clone left the primary tumor and seeded the metastasis. Now, the cohort here is different. We didn't have the luxury of having multi-region sampling from these largely publicly available data sets. So we turned and developed a very simple mathematical model that's detailed in this manuscript, but we were also able to calibrate it against our more sophisticated spatial computational models. And the takeaway from this is, again, fairly profound, namely that when we estimate the timing of metastatic seeding for colorectal, lung, and breast cancer, we see that by and large, seeding is occurring quite early, on the order of 4 years prior to diagnosis of the primary tumor in colorectal cancer, 3.5 years in lung cancer, 2.7 years in breast cancer. And I'll just point out that there's, in fact, greater spread in breast cancer, and this actually tracks with the subgroups of disease and so forth. So again, we're really finding evidence that metastatic seeding can occur early, and I think that when we put this in context, we need to be very careful about the language regarding early in light of the fact that this evolution is taking place over decades, really over a lifetime. And so the perspective of timing depends on really where we're looking at this from. Whether we're looking back in time from out here, then we're saying this is a relatively early event in the history of this tumor. If we're starting from a normal cell, the zygote, that landscape looks different. And I think that I just want to emphasize that there's a real window of opportunity to potentially be intervening earlier here. Now, we can take this sort of a step further and ask, well, how do we calibrate this, and what does this mean when we think about sort of detectability and clinical outcomes? So we can use the fact that a number of metastases occur synchronously. This is particularly the case for colorectal cancer, where we often see a patient presenting with synchronous liver metastases. And indeed, as we might expect in patients that present with synchronous metastases, they tend to have been seeded earlier than those with metachronous disease. Of course, this also depends on tumor histology. And we also see that the timing of dissemination correlates quite well with the time span from diagnosis of the primary tumor to metastatic detection. And so we think that this helps frame this within the clinical light, and is part of the reason why we focused on quantifying this from the sort of number of cells perspective. Now, if we come back to the question of monoclonality versus polyclonality, this too has been a real challenge and a really hotly debated in the field. And the scenario that I'll point out is, so we just have a single cell or cluster of cells that are seeding the metastasis that have a common genetic origin, or perhaps we have distinct clones that are seeding the metastasis. The way that we sought to tackle this question was to, again, turn to our simulation framework, where we understand the ground truth data. And so within that kind of model, we can simulate these different scenarios, and we can then look at how the patterns of mutations appear. And what you can start to see from this is that in a scenario where we've simulated monoclonal seeding, we in fact see that there's a much higher degree of metastasis-private events. So these are events that are occurring uniquely in the metastatic lineage. And in contrast, when we simulate under a scenario of polyclonal seeding, we see that there's a much greater degree of sharing here along the diagonal. So mutations found in the metastasis are also found in the primary, and this occurs really along the whole range of allele frequency values. And so you might argue that we're seeing an enrichment for what is called shared subclonal events. So many of these are down in this range. So with the simulation framework, we can then establish cut points, derive those empirically, and then ask, how does this look in the actual patient data? And so what we see is that, in fact, there's a much higher degree of polyclonal seeding in lymph node metastases. And in fact, when we look at untreated distant metastases, we also tend to see a slight enrichment for polyclonality, whereas those that are treated tend to be much lower. And I should point out that by and large, most of the lymph node metastases are untreated. They're resected at the time of surgical resection of the primary tumor. So this implies a scenario where perhaps most of the time we actually have polyclonal seeding of the micrometastatic lesions, but that by the time we apply therapy, we've actually clonally selected for this resistant subclone, and that's what comes to dominate the population in the macrometastasis. So this leads us to a model where I think we, as a community, need to really carefully think about how profoundly treatment can actually remodel a tumor's clonal architecture. And in this scenario, we imagine that we have this primary tumor that's quite genetically diverse, and that's supported by many studies from our group as well as others, when we've looked deeply enough, we see that these tumors are just incredibly genetically diverse. We might have a scenario where disseminated tumor cells originally give rise to these micrometastases, and that, in fact, when we apply therapy, we are potentially selecting for those clones that harbor pre-existing resistance-conferring mutations. There's enough genetic diversity to provide a very rich reservoir for that. And so this is what we select out for. And in contrast, of course, many other cells will go on to die. They will, in fact, be targeted by this therapy. So I think that, this is maybe a hard message to hear, but we really need to better understand the implications of the timing of adjuvant therapy and what specific therapies do to the genome. And unfortunately, with this cohort, despite being really one of the largest cohorts of paired primaries and distant metastases to date, the treatment information is inadequate to go back and resolve how those therapies impacted the genome. And that really requires very detailed, ideally dosing information, but also ideally whole genome sequence data to read out those mutational signatures. So in summary, what I've shown you so far is that the seeds of metastasis are commonly sown quite early in epithelial tumors. We estimate this is on average 2 to 4 years prior to diagnosis. We do find, and I didn't dwell on this, but relative to comparisons of primary tumor growth and pre-neoplastic growth, that the course of metastasis is actually selectively relaxed. And that is very consistent with a model where the tumor already has what it needs to propagate, not only to invade, but also to metastasize. We see that treatment really dramatically remodels the clonal architecture of the tumor, and in fact that these untreated lymph node metastases and distant mets are often polyclonal. And I suspect that we are dramatically underestimating the prevalence of polyclonality because most of the patients that we see and sequence have seen prior therapy. And then what I haven't spent too much time on, and I think we really require new tools to address, is why we don't see metastases more often. If dissemination can occur exceedingly early when we have the right combination of events, why doesn't this happen more frequently? And I really, I think there's interesting opportunities, particularly with new spatially resolved techniques, to start to probe the contribution of immune surveillance across the continuum of disease. And then finally, of course, dormancy is a key question here, and one that I think the next section that I'll speak to will illuminate. But I do want to point out that these models that I've described that are based on mutation accumulation are not ideally suited to account for dormancy. And we can modify them and think about this in different ways as we start to understand those parameters, but they are reading out the proliferative history based on the mutations that are accrued with each passing cell division. And so certainly we can think about the fact that breast, as I'll go on to describe, can have extreme latency and very long periods of dormancy. And this is a real challenge for the community to start to think about how we understand the switches that allow a tumor to escape from dormancy. So I'm going to switch gears and just in this brief last part, talk about other work that we've pursued to try to understand the dynamics of breast cancer relapse using very different approaches and very different cohorts. So many of you will know that we've seen really dramatic improvements in breast cancer patient outcomes in recent years. However, it is still the case that some 20 to 30% of patients recur at distant sites. And as of yet, as a community, we've really struggled to predict the patterns of relapse, both temporally and spatially. There's been very important work that's been done, in particular this meta-analysis by Canonical that demonstrated that, in fact, there is a subset of women with estrogen receptor positive breast cancer that have a persistent risk of recurrence up to 20 years after diagnosis. And this includes women with lymph node negative disease that would otherwise be thought to have a relatively low risk. And so this really harkens to the critical need to identify tumor features that are more predictive of the risk of recurrence than our standard clinical covariates, such as nodal status, tumor size, and grade that we tend to incorporate in these models. And what this really highlights is that there's a critical need to have clinically annotated cohorts with very long-term follow-up and detailed molecular data. And so I was fortunate to work on one of these cohorts, namely the METABRIC cohort, or Molecular Taxonomy of Breast Cancer International Consortium. And this cohort consists of over 3,000 women derived from 5 different tumor banks in the UK and Canada who were diagnosed some years ago, between 1977 and 2005. And what that means is that we now have very rich and long-term clinical follow-up, actually with a median of 14 years, up to 20 years. Now, the genomic data from this cohort, we previously reported on various aspects of both the copy number and mRNA landscape, microRNA landscape, and mutational profiles. But what I want to focus on today is that we've been able recently to go back and obtain information on the first site of relapse for all patients in this cohort, so about half of them relapse, as well as the dates and sites of relapse for 60% of the patients from one site. And so that's really unprecedented data in that it allows us to understand their relapse trajectories in a way that we haven't been able to before, because this information is not typically recorded in the SEER registries. So what we previously described with this cohort, just to recap, this was some years ago, we used genomic data and transcriptomic data in unsupervised fashion. And I want to emphasize that this was completely without regard to the clinical outcome, to discover new subgroups of disease and by combining this information. And so shown here, what we see is that we can look along the chromosome for these some 10 different subgroups. We've defined them as the integrative subgroups. We've compared them with the intrinsic or PAM50 subgroups. And what I want to point out is that these tumors are largely copy number driven, and that really hadn't been fully appreciated at this point. But I'll point out the well-known HER2 oncogene shown here on chromosome 17q. You can see this lower track plot in black shows the strength of the association between that alteration and the subgroup, and you'll see that it's incredibly significant. However, I want to say that there's a number of other groups that have very similar patterns. And this was our really first appreciation that there's subgroup-specific drivers, copy number drivers of other subgroups of breast cancer that mimic the patterns we see for HER2 positive disease. And to show you the outcomes just briefly with this Kaplan-Meier plot, you can see that these subgroups that we discovered in this unsupervised way have very different clinical outcomes. This group here in sort of brown is the IC5 HER2 positive group. So this group is very much enriched for this. It actually dramatically improves the stratification obtained by the expression-only or PAM50 groups. Very poor outcome. Of course, this outcome would look dramatically different today because we have the monoclonal antibody trastuzumab, which has been very effective in this population. But you'll see that these other groups, some of which have very, very steep mortality trajectories. So this was all fine and well, but we knew at the time that, in fact, our current approaches to survival analysis were really somewhat suboptimal for this task. And part of the reason is that they don't account for baseline hazards differences across patient subgroups and how those change over time. There were also limitations in really modeling this interdependency between the patient's clinical course, namely the fact that they would often have surgery and go on to develop a local regional relapse and or a distant relapse. And additionally, they don't adequately address the issue of competing risks of mortality, namely that ER-positive tumors tend to be diagnosed later and at a much steadier rate than ER-negative tumors. And so Oscar Urita, who is at the University of Cambridge and has just started his own group, but during a sabbatical in my lab, we worked to develop a new model to really account for these dynamics. And this, I won't delve into the details, but this is a very flexible Cox clock reset multi-state model. And you can see that it allows us to flexibly model these transitions of a patient going from having their surgery to either a local regional relapse or a distant relapse. We can then go on to model whether they die from cancer or perhaps from other causes. And so really, this is a very flexible approach and can be extended to account for other factors such as treatment. When we apply this to the molecular data set that we had in hand, we see several quite striking patterns. So in this fairly complicated plot, what I'm showing is the probability of relapse over different time intervals, so up to 20 years for each of the integrative subgroups. They've been ordered by their risk. I'll focus first on the HER2 positive group, which has the highest risk. The black triangles denote the risk of relapse after surgery, In red, we see the risk after being disease-free 5 years, and in green after being disease-free 10 years. And so really dramatically very high risk of relapse before the routine use of trastuzumab. And so we expected this to fall out on the extreme end, but what we were surprised by is that this approach also delineated 4 subgroups of what we call high risk of relapse ER-positive, HER2-negative disease. You can see that their risks exceed 40%, even well beyond, and continue to rise. So in some cases, we see that the risk of relapse is rising up to 20 years out. Adjacent to this, we see 2 groups of triple-negative breast cancer. So the IC10 group is sort of represented by the classic basal group, and we see that their risks plateau after 5 years. In contrast, this IC4 ER-negative group continues to rise, so the risk continues to rise. And these 4 groups over here would be called the more typical or average risk ER-positive, HER2-negative groups. So with this information, we asked, well, could we improve the prediction of distant relapse? And what would it look like if we compare our most powerful clinical model that incorporates all of these features, as shown in this green triangle, versus a model that incorporates integrative subtype information? So you can see that really these green lines homogenize the risk between these 2 triple-negative subgroups. In contrast, when we incorporate integrative subtype information, there's really a dramatic difference. And this group truly does plateau in its risk after about 5 years. We can do the same thing for the high-risk ER-positive groups. So these are the 4 groups over here compared to a typical risk group. Again, when we just incorporate our most powerful clinical model, the risks are homogenized not only across groups, but to some extent between these time intervals. And in contrast, the incorporation of integrative subtype information shows us that the risks of relapse really differ over these time intervals and between these groups. So Jose Sione in my group, who's now going on to start his own group in Barcelona, has built a meta cohort that allowed us to validate these findings to the best of our ability. Now, this was quite challenging because getting 20 years clinical follow-up and gene expression data is actually non-trivial. But what you can see in this scenario is that we compare our predictions using the clinical model or the IHC model versus the integrative subtype model, first in the METABRIC cohort over 10, 15, 20-year intervals. Where we trained the model was in the METABRIC cohort. We can then compare the predictions, and this is looking at the C-index, so a measure of the predictive utility, and we see that pretty much across the board, the integrative subtypes improve our predictive utility, and this is especially the case when we look 20 years out in these late relapsing ER-positive, HER2-negative groups. So importantly, these groups actually also contain specific driver alterations. And so just looking here, I want to emphasize that we believe we've identified the quarter of the ER-positive, HER2-negative population who are really at high risk of relapse. And each of these groups are broken down by the proportion of ER-positive cases that they account for. So in this case, 8%. And I'm showing you here the amplification landscape here at chromosome 17q23. So this is to say that in this subgroup of cases, almost all of them exhibit amplification and as well as overexpression of the genes at this locus. And of course, one of the key challenges that we have is pinpointing the specific drivers since there's many genes in this amplicon. So we can go across the board and see that this is similarly true of the IC6 group with amplification overexpression of FGFR1 and ZNF703. IC9 is a bit more complicated. It turns out this group harbors very prevalent amplification of MYC on 8q24, but other genes as well, such as NCOE3 at the 20q13 locus. And then we have this very complicated architecture where we see that for IC2, they co-amplify not only the CCND1 locus and the FGF ligands, FGF3, 419, but they also exhibit amplification of this cassette of oncogenic drivers that are really implicated in chromatin regulation, namely RSF1 and MZ. So we believe that actually there's new avenues to therapeutically target these subgroups that are at highest risk of relapse, and this work has led to trials to now investigate that. So just coming back to this cohort, I do want to touch on the fact that really we have very comprehensive information on relapse now at our fingertips. And so this summary plot, just looking at ER-positive versus ER-negative disease, shows the sites of metastasis that we observe in these patients. And as you can see, irrespective of hormone receptor status, these tumors metastasize to many sites throughout the body. And often more than one site. And so with this information, we can really now begin to delineate the rates and routes of metastasis, as well as their associated lethality. And I think it's important to say that many of these subgroups, including those with the most favorable prognosis, such as integrative cluster 3, which we often think of as a typical risk group for comparison, in fact has often more than one site of distant metastasis, sometimes 2 or 3 or more. And so that's really important as we understand the etiology and chronology of breast cancer. And then furthermore, we can start to break this information down by molecular subtype. And we're obviously underpowered when we get to the 11 integrative subgroups, but just looking at ER positive versus ER negative disease, we can see some immediate patterns. So it's been reported for many years that bone metastases are more common in ER positive disease than ER negative disease. It's estimated, on the order of 71 versus 43%. If we call this out, what's intriguing, we certainly see that difference in proportions, but actually they have a similar cumulative incidence. And this starts to open up questions as to why we see these kinds of site-specific tropisms by ER status. And given that many of these ER positive patients often develop a bone met first, this may potentially explain it. It's also the case that bone mets may take a number of years to develop, and so this is less commonly observed in the triple negative cancers. Now, the other thing that I do want to emphasize is that with this information, we can now examine the impact of these different factors on a patient's log hazard ratio of survival. And just broken down by ER positive versus ER negative disease, what I want to point out is that once we start to incorporate additional information about a patient's prognosis, we can see that these original covariates, such as lymph node status, size, and grade, really lose their predictive power over time. And so that's very important as we try to build better models to understand these dynamics. So in summary, really we find in this unprecedented cohort that the risk of relapse and the timing of first as well as subsequent relapses differs across the major breast cancer subgroups. We find that the integrative subgroups can improve the prediction of late distant relapse compared to standard clinical covariates. And we have now identified the subgroup of ER positive patients that are at highest risk of relapse. And we believe that there are new strategies to target these women and are very excited about harnessing this approach to improve patient stratification. So I'll just close by saying that I think I spoke a little bit about this, that, so far I've just talked about genome sequencing and gene expression data, and there's obviously many other tools at our fingertips that allow us to start to characterize these lesions at greater resolution. And there's ongoing efforts within the Human Tumor Atlas Initiative. This has been a major focus of our group. And I think looking forward, we're really excited about how we can start to harness these in situ profiling techniques to better understand the tumor microenvironment, and really to put this in the perspective, not just of an evolutionary context, but actually of an ecological perspective. And so I'll just point out this perspective piece that emerged from an NCI workshop organized by Dan Gallahan and NASA here, that I think has, it's something that we're very excited about envisioning the next steps for. So I'll close. You've seen the people along the way who have contributed to this work, and very thankful to our funding bodies and our collaborators locally and abroad. And I'm happy to take any questions.
Thank you for a very interesting and very comprehensive talk. I know that we have some limited time for questions. I just want to remind people that Christina's slides will be available to view on the SeekSpace website, and that she also has several nice publications on the topics she presented today. If you have questions, please go ahead and put them in the Q&A box on the right-hand side of the WebEx panel. In the meantime, you mentioned a little bit some of the future directions that you were thinking about. I wonder if you can expand on that. For instance, will you be able to evaluate more aspects of the tumor microenvironment that might contribute to occurrence of metastasis? For instance, immune response or presence of microbial species? And are you looking at things like germline genetic variations, or how host factors might impact the appearance of driver mutations or metastases?
Sure. Yeah, it's a multifaceted question. I think we're interested in many of those things. I think obviously the microenvironment has been largely overlooked, and I point that out in part because with these spatial models that we've been developing, we think that's a very important component. And we do, are beginning to have the tools necessary to start to probe tissues in situ, whether that's at the transcriptomic or proteomic, and to maybe a lesser extent today, but hopefully more so in the future at the genomic level. So absolutely, I think we've been really trying to bring forward some of those new technologies and develop new computational approaches to interpret them, and have done so in the context of a number of clinical cohorts. So I think that's, it's early stages, but we're beginning to learn. You asked questions about the germline, and certainly that's a component that we're very also interested in. It's been the case that I think a lot of these cohorts haven't necessarily had as much detailed information on germline or epi data, but we're really, looking forward to bringing in some of the cohorts that are becoming available with somatic sequence data and gene expression profiling. That, I think will be a very interesting opportunity, and looking forward to what can be accomplished there, both in terms of the extension of the models, but really, I think just, speaking to the fact that many of these events are happening early, understanding their context dependencies with respect to the germline. I guess I can say that one of the ways that we're doing that is through the Human Tumor Atlas Network Initiative. There's several different projects that are under that umbrella. One of them is a precancer atlas of familial adenopolyposis. And so with these patients that harbor germline FAP mutations, there's a massive effort underway to now characterize their genomes, transcriptomes, metabolomes, pretty much every ohm you could name. And I think that that's going to be a really rich cohort to try to learn the relationship between germline and somatic variation.
Yeah, and as you say, it can be difficult to get the studies together that have all the types of data and longitudinal information that you want on treatment, as well as materials available. So it's nice to see that studies like yours are being able to address some of these questions with some decent sample sizes. So we have a couple technical questions probably I'll ask before wrapping up. In the first part of your talk, I think there was just some questions in terms of, I think you mentioned you did exome sequencing and also targeted sequencing. Were those both tumor and paired normal samples in both instances?
That's right. So in all cases, we had a tumor and a matched normal. Because these were, we were really seeking cohorts with very strong clinical annotation. In some cases, an FFPE normal was used from a distant site because blood was not available. But yes, absolutely, to robustly call somatic mutations in those studies, we required some normal sample. And for the first study, in fact, it was all based on exome sequencing, and same for the second study looking sort of pan cancer.
And then another technical question, for the sequencing data, what type of data did you look at? For instance, was it indels, SNVs, copy number alteration, hypermutation status, or was it really just, a variant, SNV?
Yeah, so no, it's a great question. We looked at, predominantly focused on SNVs as being the majority, and trying to compute their prevalence in the cell population, so the cancer cell frequency. But we also absolutely look at indels and copy number variation, and in fact use the landscape of copy number variation to then adjust those allele frequencies. And that's really quite critical to getting these inferences correct, because many of these tumors are aneuploid. Sometimes they are triploid, quadruploid, but also the local copy number variation can alter the allele frequencies, right?
Thanks again. So we're getting close to the end. I just want to thank you very much again for a very informative talk. I think people really appreciated hearing your expertise and experience. I'm going to turn it over to Leah to wrap things up.
Thank you.
Great. Thank you, everyone, for joining us today. And thank you, Dr. Curtis, for a really fantastic presentation. We really appreciated it. And I wanted to, we welcome everyone's feedback to inform future webinars. Lastly, just to announce, please stay tuned for future webinars. We are starting back up for SeqSpace in 2021, and we'll be updating our website and with registration information soon. So thanks everyone for your participation today, and have a great rest of the day. Thank you.